This video will survey Pathway Tools microbiome data management capabilities, in particular some of the tools we've developed for helping a scientist decipher metabolic relationships between organisms in a microbial community. I'm going to start out by showing tools we've developed for issuing queries across a set of metagenome assembled genomes, MAGs, from organisms in a microbial community. Now here I've got a number of PGDBs in my startup screen. Now normally in Pathway Tools, you have a single organism selected that all your queries are issued against. But there's a new mode in Pathway Tools where you can define a set of organisms as your query set. To do that, you click here, you say define a new organism set, and then we're going to select in the left pane all the organisms that we want to have in our organism set. And in particular, we're going to select the 8 organisms in the altered Shadler flora. We'll give that organism set a new name, ASF, and we'll create the organism set, and we'll say, okay, that's our current selection. And Pathway Tools now opens all those databases. And so these are the organisms that we just selected, and they'll be the subjects now of any search that we run. First search I'm going to do is to search across all these organisms for which organisms have the compound spermidine. And so we see that 4 of the 8 organisms contain that metabolite. For one of the organisms, there are 2 reactions involving 1 metabolic pathway. For the other 3 organisms, there's 1 reaction each involving 0 pathways. And if we click on that organism, on spermidine for that organism, we'll see the reactions and the pathway that it's in. Notice one of the reactions is a generic reaction not involving exactly spermidine. That's why only 2 reactions were listed previously. Now let's go back. We can also do a protein name search, so I can search for pyruvate kinase across all these organisms, and we can see that most of the organisms have pyruvate kinase. And in this smart table, it's showing us the protein, the gene coding for it, accession number, which organism it's in, the length of the gene, and then the surrounding genome neighborhood. We can also search for pathways. So I'm going to do a substring search for pathways. I'm going to search for tryptophan biosynthesis, see which organisms have that. We see 3 organisms have tryptophan biosynthesis listed here. So I can click on one of them to see the pathway in just that organism. And then I can ask for a species comparison across. I'm going to select all the ASF organisms. And we got a table summarizing which organisms the pathway is present in. It's actually present in this one, but not present in this one, even though 2 of the enzymes are present. The full pathway was not predicted as present because there was too little evidence. And we see the operon organization as well in each of the organisms containing it. Let's do another comparison now between organisms in this community. Let's actually click on one of the organisms to select that as our current organism. Let's show its metabolic network, and let's now do a species comparison of that metabolic network. We're going to do the comparison with this lactobacillus species. And now everything highlighted in red is common between the 2 organisms, and the pathways still in blue are present only in this Clostridium and not in the second organism that I selected. Another thing we can do is we can build a community cellular overview diagram consisting of metabolic networks for several organisms in the community. In this case, we're seeing 6 of the organisms with their entire metabolic networks present. And one of the analyses we can do here is to do a species comparison where we highlight either the reactions that are common to all the organisms or the reactions or pathways that are unique to one organism. So here in red, we're seeing all the pathways that are unique to each organism. Now, the final tool we're going to look at is a comparative pathway analysis between all the organisms in this community. And we invoke this, although this looks like the Biopsych website, this is our own local pathway tools web server. We're going to go under the comparative analysis menu, and we're going to select a pathway analysis. And for the organisms, we have all of the 8 altered Schadler flora organisms selected as our organisms, and we're going to submit that query. And now we get a pathway comparative analysis result where what we're seeing are analyses across the columns in this output table are all of the organisms, and the rows in this table are different classes of metabolic pathways. So in this first row, it summarizes the number of biosynthetic pathways present in each organism. And then the subsequent rows break down the number of subtypes of biosynthetic pathways: amino acid biosynthesis, tRNA charging, aromatic compound biosynthesis, etc. And then here are all the utilization pathways, energy metabolism pathways, etc. Then we get total pathway counts across all the different organisms. And if we want more detail on any of these rows, we simply click over here. And here, for example, I clicked on the amino acid biosynthesis. And so I get a summary, again, across all the different organisms of specific individual amino acid biosynthetic pathways present in those organisms. And here I've clicked on cofactor biosynthesis, and we get a summary of all the cofactor biosynthesis pathways present in each organism.